organized in the conference hall, second floor, Faculty of Science. So if people want to join there, they can also go. Thank you. Hello. Good morning. Namaste to all present over here. I welcome you all to the second day of the International Conference on Integrative Chemical Sciences for Health and Environment. So, we all have gathered here from different places. Some are from Kerala, Udaipur, USA. So, it's not only about the talk on science, it's also about exchanging emotions, feelings, and also science. So, with the permission of the chairperson, I would like to begin the session. So, first of all, I would like to introduce our chairpersons present over here. I would like to begin with Dr. Rama Shastri. He obtained his PhD in chemical sciences in 2005 from the Department of Organic Chemistry, Indian Institute of Science, Bangalore. He then pursued postdoc at the Scripps Research Institute, San Diego. He joined as an assistant professor in 2011 and then associate professor in 2017 at the Department of Chemical Sciences, ICHE Mohali. So his research interests are very wide, which includes palladium catalyzed allylic alkylation reactions, one post cascade transformations. So a list is long, so I would directly jump onto the awards. He's a recipient of CTRI Award for Excellence in Drug Research 2022. He is also been awarded with RSC Research Fund Grant in 2020. He is also OPPI Young Scientist Award 2080. He is also under the Leaders in the Field uh, LITF scheme. He also served as an editorial advisory board member. So a list is long. I would like to welcome Rama Shastri sir to our conference and to our college. So the next year person of our session is Professor Venkat Krishnan. He completed his PhD in physical chemistry in 2006 at University of Stuttgart, Germany. He also worked as postdoctoral researchers at University of Pennsylvania, USA from 2006 to 2010. Worked as research associate at National Institute of Material Sciences, Japan from 2010 to 12. Presently joined as a faculty at IIT Mandi in April 2012. He works in the field of green chemistry and heterogeneous catalysis for energy and environmental applications. Currently guiding 7 PhD and 5 MSc students. He has published more than 170 articles. He has also been bestowed with several awards like DST Inspired Faculty Award, IIT Mandi Foundation Day Award for Excellence, DAD Visiting Scholar Fellowship. So many more things to his CV. So without wasting much time, let's start the session. And I would like to call Professor Rama Shastri and Professor Venkat Krishnan to please start the session. Over to you, sir.
Good morning, everyone. Uh, let's get started with the first session. The first talk of the first session will be delivered by Professor A. Saktivel. Let me introduce Professor Saktivel to the audience here. So, Professor Saktivel has a teaching experience of more than 21 years. He is presently working as a professor in the Department of Chemistry, Central University of Kerala. He has a research experience in materials chemistry, development of eco-friendly heterogeneous catalysts for biomass, fine chemicals, and petrochemical processes, and also development of eco-friendly heterogeneous catalysts for various other purposes as well, including energy, environment, and healthcare applications. He is a recipient of many awards, which include Sistla Kameshwari Young Scientist Award, Maya Devi Juneja Endowment Medal, and he also was a recipient of the famous Alexander von Humboldt Fellowship. He has authored 5 patents, 3 internal reports while his tenure at Reliance Industries Limited, and he has several papers and more than 130 research papers. He's a member of many academic society. He's a fellow of the Royal Society of Chemistry. He's also a member of the American Chemical Society and also the International Zeolite Association. Without much further ado, I request Professor Saktivel to deliver his talk. You'll have 20 minutes for your talk and followed by 5 minutes for the question and answers.
25.
25, it's fine.
25. Thank you, Professor Venkat, for your kind introduction. First of all, I take opportunity to thank the organizing committee for the giving opportunity, and also thanks for nice arrangement. So today I will be talking about how the heterogeneous catalysts, in particularly zeolite-based catalysts, are sustainable for the converting biomass model component to value-added chemicals. Because the talk will be present, I'll be sharing with why we need to go for the biomass component, and then what is the importance of zeolite in its application. All these aspects we'll be covering. So before going to the talk, I just want to give a brief introduction about our institute. Uh, Currently, I am associated with the Central University of Kerala, which is located in the north part of the Kerala, extreme north of the Kerala. It is in the border between the Karnataka and Kerala. The nearest airport is Mangalore and Kannur. From here 2 hours, and we have 300 acres beautiful campus. The campus established in 2016. We have moved the new building 2018 Department of Chemistry. We have the reasonable good infrastructure, and we have master and PhD program. So those who are interested, just you can go around the website and find the more details. So coming to the talk, so why do you need a renewable feedstock? What is renewable feedstock? Because we all know that there is a deflation of fossil fuel, and we are going to have the another 50, 60 years. Then we are going to really need a energy alternative chemical methods for the day-to-day life. So it is important to find renewable feedstocks. So some of the feedstock here is like a forestry wood moss, agricultural biomass, and then animal residue and municipal waste. These are all important residual biomass component which can be useful for the as a renewable energy source as well as for the chemical also. And other than this biomass, there are also hydrogen economic and carbon dioxide reflection. These are also important aspects one can think about renewable resource. Why do you need a renewable resource? Because we have a increasing demand in every year. So you can see that from 2019 to 2021, from 805 to 1400, the billions of units energy requirement is there, and this energy requirement has been fulfilled by oil source, natural gas, and coal. These are some of the energy resource currently used, and we can see that with the help of this energy resource, we are producing a lot of carbons from. So we can see that China, US, and India, we are all producing a lot of carbon resource we are producing, waste we are producing. So it is important to find alternatives resource for the future renewable resource. So for that, see, if you see that in 2026 we need around 25% of utilization by using the chemicals, and energy will be utilized with the help of the renewable resource. So if you see the even 2030, 1/3 of all the chemicals and the energy resource will be produced from the renewable resource only. So this is a most important component. We have to focus on that. In this regards, you see that biomass is the one of the most important renewable resource, like in particularly agricultural residue, forest resource, organic waste. So which can be first with the help of pyrolysis and other hydrothermal treatment, we can get a sugar and intermediate building blocks like gaseous mixtures. We can get it. These gaseous component can be converted into valuable added chemicals like fuels and chemicals, which is an alternative for the what you are getting conventional chemical from the petrochemical process. So often these methods from here to upgrading process always involve the catalytic process. So here is some of the examples of biomass component to value added product. For example, levulinic acid to ethyl levulinate. It is a fuel additive directly used to the blind with diesel and petrol, and vanillin is one of the important intermediate chemicals. They all produce from the biomass component directly by the conversion. And often another important example is when you take about the biomass component, lignin is one of the most important biomass component and which has a lot of phenolic components. So the phenolic component have a lot of oxygen and aromatic hydrocarbons are there. So this aromatic hydrocarbon can be converted to value-added products, one. And other thing is when you hydrogenated these aromatic hydrocarbons and phenolic compound, so you can get a the precursor for the fuel. That also it's important. And most important thing is biomass components are having the oxygen rich, always oxygen rich will be there. So it is always important to deoxygenation by the hydrotreating process. So those are the like hydrotreating process is one of the important process in the biomass to value-added chemicals. So often these kind of reaction, for example, if a ethyl levolinate synthesis or other dehydrogenation reaction, which are often involved with the sulfuric acid or even the fluorinated compound and various other. Mostly, if you see that fluorinate, paratoluene sulfonic acid or sulfuric acid, mostly it's like a corrosive chemicals are used. So it is important to find the alternative substituted catalyst. It is important. So in this regards, the heterogeneous catalyst is the one of the most important catalyst. So because we can recover, often we can recover and reuse it. And another thing is even petrochemical industry, because petrochemical and biochemical refinery, it is very important to use the zeolite or inorganic materials because they can run for continuous process. So there will be a less effluents going to get it. So it is important to develop the heterogeneous based catalyst. So here we are going to focus on the zeolite and zeolite like molecular sieve based heterogeneous catalyst. We are going to talk about it. And zeolite source comes into the porous materials. They have been classified into three different categories. You can see that the normally the porous material has been classified into microporous, mesoporous, and macroporous. The zeolite comes under the category of microporous materials, and it has a lot of channels and voids. The major advantages, you can see that the zeolite normally have the solid acid. So it can have the tunable acidity, one can tune the acidic strength and the acidic property, and it has a good high surface area. And as you can introduce the redox center, like whatever the redox center you can have it, you can introduce into the framework or non-framework matrix, which can have the potential application. So for the students' purpose, so they are all like zeolites are derived from the silicate, aluminosilicate derivative. So they build with the different architectures, and they can get a different type of porous architectures, and it has a different surface area, and also it has a different pore dimension. We can, as based on the nature of the starting material, we can tune the property of the structural property. So we all know that zeolite has been excellent material for gas drying and also water purification, as well as carrier for the biomass molecules, as well as petrochemical industry. It has been extensively used in. Particularly, we are enjoying the gasoline today because of the revolution derived from the zeolite chemistry, because FCC catalysts all derived from the zeolite based catalyst. So, and as I mentioned that we can incorporate different metal ions. So if you want to introduce a redox centers, you can introduce redox center. If you want to introduce a acid based tune property, you can also introduce. So that is the advantage of the zeolite based chemistry. So what we are doing is we are trying to develop the different types of zeolites and then try to explore that like hydrogenation of lignin based molecule and conversion of lignin based molecule into value added product like vanillin, and also hydrogenation of aromatic hydrocarbons. Then also we are exploring the CO2 utilization and other. These are the different area of research we are working out with the help of the zeolite like materials, and also we also working on the mixed metal oxides. Basically, we two types of material we are working. One is the zeolite based materials, and other one is the mixed metal oxide derived materials. So here I am, because of the time constraint, I will be restricted to zeolite based matrix only. So we'll focus on the different types of zeolites which we are focusing. So mainly we are focusing on the large pore and high surface area zeolite like ITQ, zeolite beta, and MCM22 type zeolites. Because these zeolites have the unique property. I will talk about later what are the properties. So, and we try to introduce the heteroatoms like oxidation ability we tune, and then acid base ability we tune, and also redox property all tune, and explore the biomass model component to value added chemicals. That's what we explored. So first let us talk about that, the oxidation of isogenol, because when you pyrolyze the biomass component, this isogenol is one of the intermediate it comes. So when you convert isogenol to vanillin, which you know that these are flavored materials, it can be potential application. So we have chosen the zeolite called MCM22. See this zeolite, it is like what we are having in the hall. It is like a uniform porous. We can accommodate molecules. And the most important is there is a black color. You can see that there is like a bowl. It's like a bowl, external bowl, where we can put whatever the heteroatom we can put it, and then it can be easily accessible. So we thought putting into the cerium based catalyst, because cerium is a high oxidation potential, that is a 1.62 electron volt, and mostly the cerium is a good desorption property. It doesn't absorb the molecules on the surface. It doesn't allow to coking. Normally it called as a heterogeneous catalyst, coking will be there. So cerium normally is used as for avoiding the coking. So that's why we have chosen the cerium based catalyst for the oxidation of this biomass model compound. So this is a zeolite we synthesize. When we synthesize, we conform with the powder XRD. So normally you can see that as the cerium is incorporation, that signal intensity is reduced, that indicate that they are in the framework. So when there is a framework is there, that it is one which plane it is located, accordingly that intensity of the signal is going to reduces, and then XPS studies confirm that the cerium is the +4 and +3 oxidation state. So if it is a completely +3 oxidation state, it is a +4 oxidation. If it is a completely +4 oxidation state, they are not good for the oxidation ability. So it should have the tunable oxidation ability. So it should have there is a switch over between +3 and +2, +4 should be there. So that we can see from here that, because zeolite have the two type of matrix. One is in the frameworks, other one is the external frameworks. So those who are in the external frameworks, there will be cerium 4+. Those in the framework, it will be mostly in the cerium 3+. So cerium 3+, partially it can be present in the tetrahedral frameworks. So that makes that the flexibility of the matrix, the oxidation potential. So, and the presence of cerium in the can be confirmed with the surface area analysis. Basically the nitrogen absorption analysis, which shows the sharp uptake. Here you can see that there is sharp uptake. It's going to the due to the nitrogen filling in the narrow pores. But then when you put the bulk cerium, so because cerium is a bulk, so when you put it, there is a decreasing the absorption. So we can reflect here also the initial, the parent material is around 487 is the surface area, which is goes to 344, which confirm that your cerium, the bulk cerium is in the channels of the zeolite molecular sieves. And then your SEM image, it is a characteristic of MCM22. It is like a bundle like structure, which is made by each layers has formed in the bundle like structures. As the cerium is the bulk ions, when you put any heteroatoms, the structure going to be delaminated. So little bit delamination is going to happen as the concentration is high, the complete delamination of the structure of the matrix. But then original structure will be remain, but then they are in the high dispersed way, that we can see from the SEM image, that confirm that matrix is very stable and it's completely delaminated. The cerium is well dispersed. That's what indication. Then we have explored this reaction for isogenol using hydrogen peroxide as the oxidant, and at the temperature is very ambient, that is 60 degree for just 1.5 hours. So we can use the different temperatures. We have studied with the different concentration of cerium. So the introduction of 5-8% of cerium help for the almost maximum of around 65% conversion with the 70% vanillin. So this reaction always the challenges other than vanillin. Always there will be a guaiacol acetate and other components, a major component will come. But then with the help of zeolite matrix, zeolite has that is advantages. It can have the selective absorption, will give the efficiently the what necessary product we need. So we can put 70% selectivity with the vanillin is the major product. And among the various oxidant, hydrogen peroxide is found to be the best. And we can see that the catalyst can be recycled for the several, at least 5 cycles. It can continue further. So we have shown that how it will be potential for oxidation reaction. Then next we choose that another zeolite to convert that levulinic acid to ethyl levulinate, because we can get a different types of alkyl levulinate. That alkyl levulinate is one of the most important components as a biofuel additive, because you get in particularly ethyl levulinate. Ethyl levulinate we can add directly to the petrol as a fuel additive. So we try to prepare this with the help of the acidic zeolites. The acidity of the zeolite can be tuned with the help of the isomorphous substitution. in particularly if you put in the framework substitution, then it can change the acidic strength. So we have tried to vary with the different metal ions like aluminum, gallium, boron, titanium, different metal ions we have tried to load and then tune the acidic properties. And then so the formation structure we confirm, and we can see that irrespective of the metal, we can see that zeolite beta is one of the large bore zeolite and having that uniform particle size with the nanocytes of in the range of around 500 nanometers. And we can see that when you have the isomorphous substitution, there will be not much change in the surface area. Surface area will be in the same range. But if you are in the external framework, there will be a surface area changes. So here also absorption also not much changes. So we get all the material have the similar absorption and similar surface area that support that we have good acidity in the they are isomorphously substituted in the matrix. So when you look into the acidity, normally ammonia will be absorbed and then ammonia TPD will be followed for the acidity measurement. So we can see that there is a strong acidity when you have the aluminum incorporation, but then when you have the zinc and aluminum is there, it has the strong and moderate acidic sides are there. So these are the different acidity, and then we try to explore for the under reflex condition, not actually 120, depends upon the boiling point of alcohol. Under reflex condition, we have studied for the alkyl levolate synthesis. So we can see that first we have optimized with the butyl levolate at 400 rpm, because when there is a rpm will tell about a diffusion limitation. So when the higher rpm there is no change in the conversion, that support that the diffusion limitation is there is no diffusion limited on the heterogeneous surface. Then we can see that it can be activity remain almost constant over the cycle. Most importantly, we try to explore the different kind of zeolite using different alcohol. For example, methanol, butanol, ethanol, pentanol, all. So when you go for higher alcohol, most of the catalyst gives better. But then the most important is ethyl levolate. But then we can see that use of aluminum beta, which is having the strong acidity, which gives only 50%. So ethyl levolate selectivity selectively yield 50% using the aluminum beta. Then of course the catalyst retain structure again remains in constant. Then subsequently what we try to do is we are trying to do that introduce a heteroatom like ruthenium or nickel or rhodium, those kind of noble metals, because noble metal containing zeolites are well known for hydrotreating in the petrochemical process. The same way, the petrochemical treatment, same way, biorefinery, it is important. We need to have the noble metal containing zeolite can be applied. So we have taken example of ruthenium based zeolite zeolite beta has been taken as a, so it is simple by post synthetic method modification. You are going to disperse the ruthenium on the surface and then make a ruthenium nanoparticles in the surface and then explore the activity. So we are prepared with the ruthenium sample and then spent catalyst which shows that there is a ruthenium nanoparticles. So it is basically initially it present the ruthenium oxide, but when we do the reaction at 110 bar pressure of hydrogen, so ruthenium nanoparticle will be in situ generated. So that's where ruthenium nanoparticle can be seen in the spent catalyst, and well dispersed ruthenium species can also see that in the from the EDAX TM and the ED pattern, electron diffraction pattern also we can see that. So and ruthenium presence of the press sample, it is a variable from +4 to +0, it's there. But then the temperature program reduction, that is the reduction ability of ruthenium, we can see that at 180 degree under hydrogen pressure, so it can see that complete reduction of ruthenium. There is a consumption of hydrogen, it comes characteristic of reduction of ruthenium, we can see that. So this material again we have explored for the deoxygenation and hydrogenation, because lignin molecule have a lot of polymeric phenolic compounds. So you remove the oxygen, because when you want a blind with fuel, we don't need to have the oxygen content, so we have to deoxygenation, and also we need to hydrogenate. So we have taken the cresol as the model compound. We have studied it. So when you have the 5-8% of ruthenium, we can see that complete conversion of your activity, and also at 170 degrees centigrade 10 bar pressure, we could able to see the complete conversion of the cresol to cyclohexane. But the problem is still, although ruthenium is very good for the coke resistance, but you can see that around 12% carbon absorption. This TGA result shows that there is a 12% carbon absorption. So that's real to the, when you do the recyclability, the product distribution varies. Initial cycle we get a methyl cyclohexane, but then subsequent cycle you can see the toluene and methyl cyclohexanol, that is deoxygenation is limited. But then when you recover the catalyst and then calcine and then reduce it under condition, we can regenerate into the original activity even after 6 cycles. So that is an interesting thing. But then ruthenium, we all know that it is expensive. So the another idea is whether can we go with the other than like a ruthenium or the precise metal, instead of precise metal, whether can we go for simple one like nickel. So that's why we try to make another important matrix, that is a ITQ. It is ITQ is a unique zeolite because it is derived from XCM22 zeolite, because this zeolite, it is by the delamination. Basically, it is kind of layer. When the layers are condensed, it forms the three-dimensional network. But when the layers are delaminated, then it can form the two-dimensional sheets of structure. That is what we call the ITQ. That is what we call the ITQ structures. So the thing is, zeolite is a layer, and can you make some metal oxide hydroxide, can you make in the layer? They can be decorated 1 hour above. Then what you are going to have it, like it is like a uniform distribution of active sites and then well exposed active sites. It can be good for more efficient for the any kind of reaction. So we have chosen that in this respect, nickel is one of the most important metal which can form the layer structures. So can basically the initial precursor will be like uniform arrangement of this layer, zeolite layer. When you put nickel, what will happen? The nickel form the hydrolyze and form the layer. So nickel hydroxide layer will form, and that will form the 1 hour above. That is what we call as a delaminated and forming the uniform layer of hydrotalcite. It is called as a hydrotalcite. So it is a zeolite layer, then hydrotalcite layer, and then when you make this kind of layer, then what is interesting is under the reaction condition, this nickel is well exposed. They can go as a metallic nickel, and that will be good for hydrogen spillover. Once the hydrogen spillover is there, it's good for the reduction. So you don't need to go for the noble metal like ruthenium, rhodium, palladium. Those are well known to use for this petrochemical industry in the huge metric tons. So we can avoid it. So that's the reason we have used this kind of ITQ, where nickel decorated ITQ type materials. So the delamination, normally we can see that there are 4 quadrant peak will be there. When there is a delamination, that peak intensity will be reduced. But then the surface area will be, when there is delamination, you can see that almost similar only. So, and then XPS shows that nickel in the +2 oxidation state. It is in the XPS shows around 861 and 884 corresponding to 2p3/2. Nickel in the +2 and +3 oxidation state. So, because the layer nickel exists in the +2 and +3 oxidation state, but when we can also see from the TEM, because this is the two kind of layer. One is a zeolite layer, and other one is like a kind of hydro. You can see that this matrix. You can see that there is nickel is decorated on the surface of the your hydrotol, like your ITQ material. So that makes that there is a good exposed active sites. So again, we try to explore for this metacrystal oxidation, metacrystal hydrogenation, because see, metacrystal is one of the simple molecule of, we are taken a model compound, but when you go for metacrystal to propyl cyclohexane, you can take even lignin model intermediate compound which can be converted into methyl cyclohexane or aromatic. That is, it's called as a naphtha. It is a naphtha component. They can be further reformed into the fuel. That is the idea. So we can see that, but here the amount of ruthenium is that amount of nickel is around 20%, where we can get a complete conversion. So 20% at temperature in the range of similar range 170, but then pressure we need relatively high pressure 20. Whereas in the case of using ruthenium, we can do at low pressure, but use of bigger nickel, it's relatively less expensive, so we can have the maximum at 28% of nickel loading and 20 pressure at 170, we can get it good conversion. Whereas the activity almost we can see that remain intact, and the reaction follow the first order kinetics. And not only metacrystal, we can see that various aromatic hydrocarbon like toluene, cumene, parasamine, phenol, anisole, all the hydrocarbons can be converted into saturated hydrocarbon. That means it can deoxygenated, it can be dehydrogenated. So that's what interesting feature. So in summary, what we like to express is that we can develop the zeolite based heterogeneous catalysts. They are more sustainable, and they are potential for replacing like sulfuric acid and other kind of because the pH what you are having, the sulfuric acid 12 pH 2, it is almost like the same in the case of your ZSM5 or ITQ or zeolite beta type. So we can replace, and then the presence of internal and external pockets in the zeolite makes that the surface is flexible for incorporating various heteroatoms, which will be good for the oxidation, reduction, as well as the acid catalyzed reactions. So I once again thank the organizing committee for the given opportunity. This is my team. So I worked in Delhi University. They were the people who worked in Delhi University. Now they are all almost settled. He is scientists and they are all faculty and they are in abroad. This is my team, current team at Central University of Kerala. So you can see that it is a top mountain view where our buildings are in the backside. So we are just in the top of the mountain. We are just standing. So this is our lab at Central University of Kerala. I like to thank DST SCRB for the funding and also our university for the opportunity, CSR and university also for the funding. Thank you once again for listening.
Good. Thank you, Professor Saktivel, for a very interesting talk. I hope there would be some questions from the audience. Yeah, please, Ram.
Yeah, a nice talk. You've shown, of course, your expertise in synthesizing different zeolites.
Yeah.
For example, ISO leveling.
Yeah, ISO eugenol.
Isoeugenol to vanillin.
Vanillin, yes, yes.
If we talk about that,
Yeah.
can you explain how your catalyst, reacts with that compound to form this product mechanism?
Yeah. Actually, basically,
here are the earlier case also you show. Yeah.
Yeah.
For example.
Basically, the zeolite matrix, the aromatic pi interaction going to happen with the zeolite matrix. When it have the pi interaction is there will be your metal center cerium will be coordinating with the olefinic centers, and then there will be a cyclic. The peroxide also going to come and interact with the cerium center and form the cyclic peroxo intermediate will come. Then there will be a cleavage will be there.
That's what. How that cleavage occurs versus what kind of cyclic intermediate?
It is that we couldn't able to confirm the cyclic intermediate, but in the case of molybdenum case, we have done with the molybdenum also. The molybdenum case, we could able to isolate the peroxocyclic intermediate. We could able to isolate. That's on the basis only we are proposing the mechanism.
Yeah.
So basically,
because my more interest is how that is cleaving. The CC bond is cleaving.
It's through the peroxo. Peroxo species will form, then followed by the cleavage. It's the peroxo species formation. Molybdenum is well good. We can give the intermediate evidence because the intermediate product also we could able to isolate. But in the here we couldn't able to isolate the intermediate we couldn't able to isolate.
Okay, thanks.
Thank you. Any one more quick question, if anyone has? If that's not the case, let's thank the speaker. Thank you, Professor Saktivel.
Saktivel sir, please stay here for a moment. Now I would like to call Professor Ramasasti sir to kindly felicitate our speaker. Okay. So there are speakers with the memento and conference kit. And in the meantime, I request some student volunteer to kindly come on stage and kindly help us to push this desk.
Good morning to you all once again. Let's move on to the second speaker of this session. The second speaker of this session is Dr. Tanmay Majumdar. So Dr. Tanmay has received his PhD from the Department of Zoology, Vishwa Bharati University in 2010, and his MSc and BSc degrees in 2004 and 2002 respectively. He's presently working as a staff scientist and head in the Center of Excellence in Vaccine Development, National Institute of Immunology. Earlier, he did his postdoc from Augusta University, Georgia, USA in 2014. and Cleveland
Some
University,
appreciation.
Ohio in 2013. He was awarded with many distinguished awards such as the Santa Cruz Biotechnology Investigator Award, Expert of FACS Canto 2 from BD Company. He has also obtained fellowship and grant from Ramlingaswamy Fellowship from the DBT, SERB, and Faculty Research Program of the UGC Government of India. He has published more than 100 research articles in reputed journals and edited books, and has also received several US patents. Without much further ado, let me invite Dr. Tanmay Majumdar for his presentation.
Thank you. Thank you, Dr. Pratibha Kumari, Dr. Srishti, for providing me this podium to show my research work. Basically, I am a immunologist, but today I show you some work where immunology and chemistry work has been related. So this is our beautiful campus. In Delhi, you won't get that many good campus. Whatever we have, it's a beautiful landscape. And whatever the work I have done, mainly it has been done by Dr. Anupam Jit Kumari. She is actually chemistry background. She has done PhD in chemistry, and then she has come in my lab to do some biology. So without her contribution, this work was never been possible. So my theme of my laboratory, I would like to tell you, because I don't do the chemistry. I work on integration of nutritional therapy with innate and adaptive immunity in infectious disease model. Mainly, we have the two immune system. One is the innate immune system, another one is the adaptive immune system, which which involves the T cells and B cells. So we work on the relation, and we work on different signaling pathway. We work on the microbiome. You know that our gut has the microbiome, and microbiome controls our immune system, and it helps in development of the immunity. So you work on the how the microbiome is playing an important role in T cell activation, and we have the several patents and all these things. So now come to the today's talk. So you know that the SARS-CoV-2 has the spike protein, and spike protein can bind with the H2. So this is the main receptor of our body which binds with the RBD domain of the spike, and in that way the virus enters inside our cell body. So, but the problem is that there are multiple cells in our body which do not express the S2. It has been shown that, like in T cells, B cells, NK cells, they do not express the S2. In spite of that, in those cells, the virus have been found. So the question is that how the virus got entered inside those cells where S2 do not express. So these are the different papers which they have shown that the virus is there inside the cells where S2 doesn't express. So there is alternative receptors we have speculated, that is the dipeptyl peptidase 4. So in MERS-CoV, earlier you know that in 2002 MERS-CoV virus has come. It made an endemic in different places, and here they have shown that the spike protein of the SMARS, the main receptor to bind with the Mars spike is that DPV4. Means the host has the DPV4 protein which can bind with the spike protein of the Mars, and in that way the Mars virus can enter inside our body. So these are the different, we have speculated the different proteins other than S2 which can bind with this spike of the SARS-CoV-2. So what we have done the blind docking of the RBD domain of the spike protein with the DPV4. So this is the blind docking we have done, and we have found that the 5 residues of RBD C-terminus domain can bind with the alpha beta hydrolase domain of the DPP4. So you can see that the glutamine can bind with the leucine, the threonine can bind with the alanine, threonine can bind the leucine, the threonine can bind the alanine, the glutamine can bind with the glycine, aspartic acid, and as well as that glutamic acid also can bind with the RGD. So from there you can understand that, there is a hydrogen domain and hydrophobic domains are formed along with the salt bond is shown, and they have the very good binding energy, which is almost minus 14.9 kilocalorie per mole. So, and we have also found that this particular RBD C terminus domain can bind with the only alpha beta hydrolase domain. The other domain like beta propeller domain, ADA binding domain, fibrodectin domain, these are remain untouched. So the DPP4 other activity was not disturbed. Only that DPP4 can, that this RBD can bind with the DPP4 enzymatic site. And here we have shown that how the hydrogen and hydrophobic interaction are formed, and it helps the stable binding in the heterodimer. So, can you press the enter? So, no, go back. So there is a video actually. A video. So we have done the simulation also, and in simulation also we have shown that this binding is very much stable. So then we have checked in biological way that whether this RBD can bind with the DP4. So we have expressed the FC RBD in the cell where H2 is absent. To nullify the any role of S2, we have made that S2 knockout cells by CRISPR-Cas9 system, and in S2 knockout cell we have overexpressed the RBDFC, and also DPP4 is already there, native protein is there, and we have checked that whether they can merge. And you can say in overlay image that RBD and DPP4 can interact. And also we have done the immunoprecipitation assay where we have overexpressed the DP4 as well as we have overexpressed the RBD, and we have pulled down the DP4 protein. When we have pulled down the DP4 protein, and we have seen that in those sample there is a RBD. So from this data we can conclude that yes, RBD of the spike protein can bind with the DPP4, and DP4 can be the alternative receptor for virus to enter inside our body. So after that, you know that DPP4 enzyme is very much high in diabetic patient. Whoever has the diabetics too, and they have the high DPP4. And whatever the insulin inhibitors we take, basically they are not insulin inhibitors, they are DPP4 inhibitors. Like sitagliptin, linagliptin, sexagliptin, viragliptin, metformin, all are FDA approved drugs and available in market, and these drugs are consumed by the patients who are having the diabetic 2, and because they consume these drugs to inhibit the DPP4. So our hypothesis was like that if you can inhibit the DPP4 using any of these drugs or in combination, that might help to inhibit the SARS-CoV-2 entry. Did you understand? So because DPP4 and spike protein of the virus is interacting to enter inside the cell of the virus, so that's why we want to inhibit the DPP4, and in that way we want to circumvent the viral entry. So here we have checked that sitagliptin, linagliptin, sexagliptin, vildagliptin, metformin, they can bind with the complex of the DPP4 and RBD, where the most favorable energy we have found that in case of sitagliptin and in linagliptin. So then we have purchased those drugs. All these drugs we have purchased: sitagliptin, linagliptin, vildagliptin, sexagliptin, and metformin. And in different concentration, we have checked whether it can inhibit the virus inside the cell. So this is the experiment with the live virus. This is the Wuhan SARS-CoV-2 virus, which has come first time in our country. So this is the Wuhan strain we have done, and we have taken the remdesivir as a control. So when you infect the cells with the live virus, you can see the virus titer. It is significantly high.
But when we have given the sitagliptin in lower concentration, there is not much inhibition of the virus replication. But when you increase the viral entry, concentration of the sitagliptin, you can see that significant amount of inhibition of the virus. Similarly, you can see in linagliptin, there is a inhibition of the virus growth when we have treated with the linagliptin. However, vildagliptin, sexagliptin, metformin, there is no changes of virus growth. That means the sitagliptin and linagliptin have shown some promise that it can be used to inhibit the virus growth. So to determine further what we have done further experiment where sitagliptin and linagliptin we have taken individually as well as we have taken in combination. And here you can see this is the virus where the cells have been infected, and you can see the high amount of virus is there. But when we have given the sitagliptin at 0.99 micromolar concentration, there is a partial inhibition of the virus growth. But as we have increased, you can see there is a significant inhibition of virus growth, which is very much comparable with the remdesivir. And when we have taken linagliptin, also it have shown the good inhibition, but in combination, it is even better than the remdesivir. So we have taken the combination of the lowest dose of sitagliptin and lowest dose of linagliptin, and we have found that it can inhibit the virus growth significantly. So we have checked the IC50 also, and here we found that sitagliptin and linagliptin IC50 is 0.46 micromolar and 2.21 micromolar respectively. Then we have checked the MD simulation with this complex with the sitagliptin, and we have found that there is a the delta G value is very high, is like minus 65.16 kilocalorie per mole, and it shown that the after by treating with this sitagliptin or linagliptin, you can show we have found that the hydrogen bond is breaking. The hydrogen and hydrogen phobic bond is making high distance, and this helps in destabilization of this complex of the DPP4 RBD complex. So here we have checked the RMSD, RG, SASA, and RMSS value of the both DPP4 and RBD, and you can check. You can see that the DPP4 RBD complex, there is a stabilization. There is a not much changes in the RMSD value, but when we have given the sitagliptin or linagliptin, there is a destabilization. So then the question is coming that okay, DPP4 we are inhibiting, but S2 is remain there, right? And the virus can enter through S2 binding. So what is happening that spike when binds with the S2 in presence of sitagliptin and linagliptin? Because again I am telling you that DPP4 is inhibiting by sitagliptin, linagliptin, not the S2. And S2 is the conventional receptor for the spike binding. So we have checked that what is the fate of S2 in presence of sitagliptin and linagliptin. And we have checked that so this is the not the sorry, this is the in DPP4 binding. So here you can see that DPP4 and RBD can bind, but in presence of sitagliptin, linagliptin, or in combination, the DPP4 RBD cannot bind. Here you can see that DPP4 is always there. In DPP4 knockout we have taken, of course there is no DPP4, but when we have given sitagliptin, here sitagliptin has been given, there is no RBD. Here linagliptin is given, there is no RBD, and where we have given combination, here also no RBD, and of course there is no DPP4, so there is no RBD. So from this data also we can check that sitagliptin and linagliptin or in combination, there is no overlay of images, where in DPP4 and RBD can overlay the images, there is the interaction. So after that, as we have checked the cytotoxicity of the drug, and we have found that both the sitagliptin and enagliptin are maintaining the very good viability of the cells. And also, we have checked the DPP4 enzymatic activity. So we have checked that whether DPP4 enzymatic activity is being inhibited by the present treatment of sitagliptin or linagliptin. Here you can see that when the virus has been given, there is a high enzymatic activity of DPP4, which is not there in DPP4 knockout cell. Of course, there is no DPP4, so there is no enzymatic activity of the DPP4. But when you have given the virus, there is a high DPP4 enzymatic activity. But when you have given sitagliptin, linagliptin, or a combination, you can see there is a reduction of the enzymatic activity. And due to reduction of the enzymatic activity, the virus growth is being inhibited. So further, we have checked that different strains of the virus. You know that after Wuhan, we have received the alpha, beta, delta, kappa, and here you can see that these are the different strains and these are the origins. And here we have checked that this is the alpha variant, this is the beta variant, this is the delta variant, and this is the kappa variant. And in all those variants, these live virus we have used, and we have checked what is the fate of virus after treatment with the sitagliptin, linagliptin, or in combination. And we have found there is a where you can see the virus load in normal cells. You can see the virus load in normal cells. However, when you treat with the sitagliptin and linagliptin, there is a significant inhibition of the virus growth. And also we have checked that the binding of sitagliptin, linagliptin in different variants of the virus. Now, as I have told earlier, that the question is that what is the fate of S2? Because S2 is there when you are inhibiting the DPP4. So we have checked that whether sitagliptin or linagliptin can bind with the S2 enzyme also. S2 enzyme works when there is a glycosylation of the S2. Other than S2, without the glycosylation, S2 cannot bind with the spike protein. And we have found that one of the glycosylated domain of S2 can be inhibited by the sitagliptin and linagliptin. And when we have checked by the confocal imaging, here this is the RBD we have expressed inside the cell. This is the RBD protein which has GFP tag. And here is the S2 protein also we have already there inside the cell. And when we have merged, you can see that yellow means the green and red are together. That means the S2 and RBD can interact. But when we have given sitagliptin or linagliptin or in combination, there is no yellow overlay of images. So that means here that RBD and S2 cannot interact. And also we have checked by immunoprecipitation assay. Here also you can see that due to sitagliptin, linagliptin or in combination treatment, there is no interaction or heterodimer formation of RBD and S2. So this is the summary of the data. The first one is strategy. This is the first report have come in the literature that other than S2, DPE4 is the alternative receptor. After that, one or two papers have come and shown that other receptors are there. So this is the first report where we have shown that no, S2 is not the only receptor. There are other alternative receptors are there. So here you can see that this is the strategy one where we have shown that DPP4 RBD can bind. So and there we have shown that the DPP4 RBD binding can be inhibited by sitagliptin and linagliptin. Or also we have shown that this sitagliptin and linagliptin or its combination can inhibit the glycosylation of S2, and that's why virus cannot enter. So this is the lab. Is a very vibrant lab, I would say, and these are my collaborators from THSTI, ILBS, IICB Kolkata, Santiniketan, and ESIC Hospital, where we'll do all the clinical experiments. And these are my support, where we get all the funds. So with that, I again thank you the Deshbandhu College, and I stop here. Thank you.
Thank you very much, Dr. Tanmay Majumdar, for a very interesting talk and also finishing it well in time. Now the presentation is open for discussion. Yes.
Presentation. Sir, I have few questions, right?
Yes.
Queries, in fact. So you have shown there are two things. One is diabetic patients.
Yeah.
Another one is non-diabetic. So a non-diabetic.
Yeah.
The
Yeah.
drugs which you have used, it was basically given to a diabetic patients. So the ACE2 receptor, the pattern of ACE2 receptors, the glycylation pattern and any other patterns, it might vary between diabetic and non-diabetic. So have you checked the ACE2 receptor patterns with the drugs for the non-diabetic patients?
Yeah, it's a
like
very
if,
interesting question. Very interesting question. Even it didn't click in my mind. So of course we should check the what is the S2 fate in diabetic and non-diabetic patients.
yes.
So first of all, yes, diabetic and non-diabetic both have the DPP4. Even diabetic has the higher expression of the DPP4. We are mainly targeting the DPP4, but in how somehow we got that it can inhibit the S2. But we have to do the further experiment to check that whether what is the fate of S2 in diabetic as non-diabetic. That we haven't checked, but definitely we have to look into.
And another is metmorphin also
It
is used.
didn't work.
See, it didn't
We
work.
have checked it, it didn't work. As I have shown for the first figure, we have checked the other drugs, like all the gliptin drugs we have checked, but metmorphin, sitagliptin, or vildagliptin, they didn't work. Only the sitagliptin and vildagliptin have. And also we have checked by simulation assay and by that their binding energy was not that good. So it means that it is not binding properly at a proper domain. That's why it was not giving the proper inhibition.
But it would have been a very interesting if this work. Basically, there is no drugs for
Yeah.
SARS-CoV-2.
So
So if
we have worked another one that the RNA-dependent RNA polymerase is the major enzyme which helps in RNA replication of the virus. And we have shown that the one of the cancer drug is sorafenib can inhibit the domain, one of the domain that is that is the neuron domain of the RNA dependent RNA polymerase. It's a kinase domain,
yes.
and it also can inhibit the virus growth. So we have this is the host based drug. We are not targeting, we are not touching the virus. We are changing the host receptors, and in that way we are trying to clear the virus. So, but RNA-dependent RNA polymerase inhibitor, which we have found as sorafenib, this is targeting the virus. So, in both the way, we have checked that it can be inhibited the virus growth.
Thank you very much. In the interest of time, we may need to move on to
Thank
the
you.
next one. Thank you, sir.
Thank you.
You may please stay there.
Sir,
We'll
please
honor
stay
you.
back. I would like to call chairpersons to come on stage to felicitate Professor Majumdar with the token of appreciation, memento, and conference kit.
Thank you. Thank you, sir. Thank you, sir. Thank you. Thank you. Thank you. Thank you. Thank you. Thank you. Wait. Please come.
Okay, it's my great privilege to introduce the next speaker, Professor Akhilesh Verma. For this occasion, I should actually thank the organizers that I got this opportunity to introduce somebody like Professor Verma, who has been known to me since I began my career, and we have been more like friends rather than any other types of relation we think of between two scientists. Otherwise, Professor Verma completed PhD from University of Delhi and pursued postdoctoral stints with well-known chemist from University of Florida, Professor Alan Katichke, in 2001, and he subsequently joined after, of course, his work at Iowa State University. He joined University of Delhi. Currently, is a full professor, uh, and he has guided several PhDs, postdocs, and also he is as I mentioned, an accomplished scientist in the synthetic chemistry domain, and regarded as one of the top-notch scientists that India has ever produced. Otherwise, his research interests span over design synthesis of new molecules, sustainable chemistry, mechanism, and also making deuterated compounds is some of the very broad research areas. Yeah, of course, obviously, he has been recognized with several awards. Some of them are, he has been the elected fellow of Indian National Science Academy. He has been the recipient of the bronze medal from the CRSI, and he is also, of course, an elected FRSC member, and he holds several other prestigious positions, part of several prestigious committees, evaluations. With this very brief introduction, I now welcome Professor Varma.
Yeah, thank you, Rama Shastri, for very kind introduction. And before starting my lecture, I would like to thank Pratibha for organizing this wonderful conference and inviting some very renowned speakers. Can you hear me? Okay. So, but in the beginning, what I could see here, there were very few students, right? And this conference is mainly for the students. So, whenever conference is there, student must give their attention to all the lectures, right? So morning 9 o'clock, very few students were here. So today what I am going to share some of the chemistry which we started in the Department of Chemistry, University of Delhi. How many are BSc students here? Raise your hand. Okay, good. So you know the chemistry, can you turn off these lights, please? Somebody. Yeah, this also. So you know the organic chemistry, or it is inorganic, organic, physical, or medicinal chemistry. It's simply based on the basic concept. How smartly you can use the basic concept of science, right? Then you can design the problem yourself and then you can pursue your research. This is what I have done in the beginning, and you will see some of my work, how beautifully we can utilize the basic chemistry. So today, what I'm going to discuss is some of the recent synthone, right, which we have come across in 2020. So if you look here, this is what we started in 2009, orthoalkanylaldehyde, orthohaloaryl alkynes, and orthoalkylene nitrile. This is the what is the recently we have come across, and by using this, we are constructing variety of useful molecules, right? And if you look these molecules, you can see you have aldehyde, you have a cyanide here, and alkyne. So the main thing is that you have to have some useful functional group which you can manipulate for the construction of useful molecules, right? And how these can be constructed, if you look here, this alkyne can be easily introduced by a Sonaga-Shah coupling reaction. If you have a respective halide by Sonaga-Shah coupling reaction, all these, you know, this starting substrate, which we can call as synthone, can be easily constructed from respective halides by Sonaga-Shah coupling reaction. And then we are looking the application of these molecules, development of sustainable method, and which is very, very important nowadays. We have to use the method which can be sustained for a longer time and should be cost effective. And what something we have come across in recently. And look here, this is a new initiative of our lab which we started in 2018. And how this has come up, when I visited ICHE-2023 in 2017, I met one of the young faculty, Siddharth Banerjee, who was well trained in mass spectrometry. And I have, consulted, can you help us? And then we together performed the first experiment of this online mass spectrometry. What is this online mass spectrometry? We perform so many reactions, right? And so many intermediates are formed during the reaction. But we have no idea whether mechanism is correct or not. So through the mass, you can trap the intermediate during the reaction. This is what is the beauty of online mass spectrometry, right? You can see here, this is a RV flask, and this is the nitrogen connected with the nitrogen cylinder, 5 psi pressure, reaction mixture is inside, it goes through the fine capillary and through the dilution with that inside the mass. This is the SDN machine of Department of Chemistry. And this experiment we have performed in 2018. And then 2019, we have made first publication on the CH activation, where we have confirmed this two copper, like it exists in two forms, copper 3 to copper 1, copper 2 to copper 0. This is what we have proved. What I mean to say here, by using this technique, one can easily identify the reaction mixture intermediate which is formed during the reaction. And if you look here, this is the picture of I don't know, it's not going, the video is there. So if you look here, there is a complete video is there, right? But it's not going through it. So this is the, here you can control the pressure of the nitrogen, and it goes through here, right? So with this pressure and the reaction mixture, this is the fine capillary which is dipped in this reaction mixture, and the reaction, when the reaction is started, then mixture goes inside the mass instrument, and you can each and every second what is happening, you can monitor. That will give you the signals on the screen which is attached through this, right? And like, if any, there are so many intermediate which are not stable, right, which you cannot isolate, but through the mass you can detect what intermediate is formed and how long it was there, right? So this was something new, and we have a lot of result using this online mass spectrometry. And for this, one need to have a dedicated mass, right? So we performed some experiment using this mass spectrometry, and recently we visited ICHE-2023 because we don't have this dedicated mass. So we visited ICHE-2023 and performed at least 6 experiments in 3 days along with my students, and we got some very interesting results. And maybe in coming time I will discuss those results. So if you look here, the cascade, one bullet more than 100 targets. So this is what is starting synthone 2010. We started journey with this, right? Orthoalkynylaldehyde and amine with embedded with another nucleophile. We have constructed a large number of molecules which are useful for a pharmaceutical point of view, right? So what is here is aldehyde and amine. You will get imine, right? And imine, then you have another nucleophile, then can attack in cascade manner, and variety of molecule could be constructed, right? This is the journey we started in 2010, and you can see these are the publication which are the outcome of this work. And another work we started in 2009 started with orthohaloaryl alkynes. And look here, what is the interesting observation? Look here. All of you know the Buckwald-Hardwick reaction. When you have any amine, In heterocycle, when you react with aryl halide, CN bond formation takes places, right? So in what observation we got here, when you have alkyne and halide and amine, the preference is attack of NH on alkyne. That is main outcome of this work. So if you have any halide and orthohaloaryl alkyne, what is the first? You can look here. The NH attacks on alkyne, that is called hydroamination, right? That is called a regioselective hydroamination, and you will get enamine. And enamine, when you have a halide in that, with a close proximity of with this, C2 annihilation takes place. This is how we have constructed some of the natural product core and other useful molecules. And side by side, if you look here, if you have a dihalo dialkynes, you will get such kind of product, pi conjugated organic material, which has been used as organic material for semiconductors. And this kind of molecule could be constructed in one go. If you have such kind of alkyne, you can take indole or pyrrole or any other heterocycle like that. And the beauty is that in this work, what we have used is in-house design ligand. 2-pyridyl benzotriazole and hydroxybenzotriazole. This is, this is the chemistry which I learned during my first postdoc with Professor Katerisky, right? So in this, what we have used is in-house design ligand and this hydroamination. And this is how this hydroamination chemistry has come. And by using this chemistry, regio and steric preferential hydroamination, right? Rather than CN bond formation, the first is the N and C bond. Hydroamination is preferred. And these variety of molecules we have constructed, right? So this is, we got from good paper. And this was not the main outcome of this work. The main outcome is this. When we perform this reaction using DMSO D6, right? What we come to know, the incorporation of D in the molecule. That, and if you look any deuterated molecule, right? They are very, very expensive. Preparation of deuterated. So what is happening? When you have any NH, potassium tertiary butoxide will generate this N negative, and simultaneously what is happening? DMSO D6 with the presence of base generate D positive in the solution, right? Which take part in formation of molecule. So now what you have in the molecule, in the reaction mixture, you have a D positive. From where it comes? When you have a DMSO D6 and a base, so you have a D positive. So when we perform the reaction of this N heterocycle, and now here what we have changed here is a thiophene. And thiophene is electron rich in nature, right? In 2 and 5 position. When we perform the reaction using DMSO D6, what we got? Incorporation of D at this vinylic position as well as N2 and 5 of the thiophene. So what we come to know, this could be a useful method for deuteration of small molecules. And another reaction which we have performed using aminoindole with a terminal alkyne, where we don't have any halide here. And we perform the reaction using DMSO D6. What we got is deuteration of both the vinylic proton, and when we use this thiophene along with this thiophene both. So if you have a molecule with the electron nature like indole, pyrrole, thiophene, purine, those can be deuterated, right? So after getting these results, you can look here. This indole, 3-methyl indole, all different kind of molecules we have deuterated by using this. And we have been granted the patent for this. And some companies are already looking forward for taking over this process. And look here, the toluene D3. The toluene D3 is used as an NMR solvent, right? So when we started with the toluene using DMSO D6, selectively you can deuterate and you can get toluene D3. in 100% yield, right? And when you have R is terminal alkyne, along with this, at terminal position also D can be incorporated. So if you look, this toluene D3 5 gram cost is $492 in sigma Aldrich, and this is 5 years before. And if you look here, the xylene, orthoxylene, metaxylene, and 1,3,5 trimethylbenzene, all the three can be deuterated with 100% incorporation of D. And when you have phenone 3, 9-10 position, and vinyl with CS3, that can be incorporated. And now look here, this cost is 15-fold. According to, if we look our procedure, right, what we have developed with this toluene D3 cost is 15-fold decline in comparison to the sigma Aldrich, right? In some of the drug molecules also we have deuterated. You can see here the Cetron, paracetamol, dispirin, pramiprazole, this all the three. So this is the best outcome of this chemistry, and this is applied. And then we have summarized this whole hydroammunition chemistry in Accounts of Chemical Research in 2017, right? And these are the paper which are